Hello everyone, this is Hadi Zafar and welcome to First Aid Explained, where I, as a medical student, try my best to explain page by page of the First Aid USMLE Step 1 2021 textbook. Today we'll be going through the biochemistry portion, pages 48 to 55. So let's begin. So here on the first page we have something called as a peroxisome. And a peroxisome is basically a membrane-bound organelle found in the cytoplasm of virtually all eukaryotic cells. Now, these peroxisomes play a key role in the oxidation of specific biomolecules. They also contribute to the biosynthesis of cholesterol, bile acids, and even plasminogens, as well as the catabolism of amino acids in ethanol. Now, Zellweger syndrome is an autosomal recessive disorder, which is characterized by the reduction or absence of functional peroxisomes, and this is usually due to the mutated PEX genes. Patients of Zellweger syndrome will have hypotonia, seizures, hepatomegaly, and early death. There is no current cure for Zellweger syndrome, and the management is entirely supportive and based on the signs and symptoms present in each patient. Then there's Refsum disease, which is an autosomal recessive disorder of alpha oxidation, which is done by or carried out through the peroxisome. And this disorder leads to the buildup of phantinic acid due to the inability to degrade it. The patients present with scaly skin, ataxia, cataracts, shortening of the fourth toe, epiphyseal dysplasia, and the treatment involves a diet limiting the intake of foods of high phantinic acid levels, found in such as dairy, beef, lamb, and other seafoods. Another treatment solution is plasmapheresis. Then we have adrenoleukodystrophy, which is an X-linked recessive disorder of beta oxidation due to the mutation of ABCD1 genes. And this leads to the buildup of very chain fatty acids. And this buildup happens in the adrenal glands, white matter of the brains, and testes. And this is a progressive disease, which starts to show between ages 2, or more commonly between ages 4 and 10. And the symptoms include difficulty swallowing, vision problems, hearing loss, hyperactivity, and aggressive behavior. And this can lead to adrenal gland crisis and even neurological function impairment, even death. Now we have something called as a proteasome, which is a barrel-shaped protein complex that degrades damaged or ubiquitin tagged proteins. Now what's ubiquitin? Ubiquitin is basically a small regulatory protein, and this is found in most tissues. And what this does is that it latches on and tags mutated or malfunctioning proteins, which the proteasome can degrade. Now, defects in the ubiquitin proteasome system have been implicated in some cases of Parkinson's disease. Now we have something called the cytoskeletal system, which is a network of protein fibers within the cytoplasm that supports cell structure, cell and organelle movement, and cell division. And the elements of the cytoskeletal system are microfilaments, intermediate filaments, and microtubules, each having their own predominant function. So microfilaments help in muscle contraction, cytokinesis, intermediate filaments help maintain the cell structure, and microtubules help with movement and cell division. For example, certain bacteria have cilia and flagella, which is basically a microtubule. Actin and myosin are found in our muscles, and they help us contract. And this is an example of microfilaments. Now, diving deeper into the microtubule, it is a cylindrical outer structure composed of helical array of polymerized heterodimers of alpha and beta tubulin. Each dimer has 2 GTP bound, and this is incorporated into the flagella, cilia, and mitotic spindles. Also, microtubules are involved in the slow axoplasmic transport of neurons. Think of microtubules as a highway within the cell itself, and proteins called molecular motor proteins transport cellular cargo toward opposite ends of the microtubule. Dynein and chysin are examples of molecular motor proteins, and dynein it transports more proteins to a retrograde through the microtubule, meaning from the positive end to the negative end, whereas kinase transports proteins and other molecules anterograde to the microtubule, from the negative end to the positive end. To remember this, ready, attack. RE for retrograde, DY for dynein, A for attack, and K for kinesin at the very end. Clostridium tetani toxin, herpes simplex virus, poliovirus, and rabies use dynein for retrograde transport to the neural cell body. Now, drugs that act on the microtubules, a very nice mnemonic is microtubules get constructed very terribly. So M for mebendazole, G for grisolfluven, C for colchicine, V for Vinsa, and T for taxanes. Now moving on. Now here we have the cilia structure, which is also another type of microtubule, and it consists of a 9 doublet plus 2 singlet arrangement of microtubules. A basal body consists of 9 microtubules. Axonal dynein, which is ATPase that links peripheral 9 doublets and causes bending of cilium by differential sliding of doublets, and gap junctions, which enable coordinated ciliary movement. So the arrangement of 9 plus 2 microtubular arrangement is seen here. Picture number B is showing us the basal body. Now, if there's a defect in the structure of these cilia leading to a defect in the movement, you can have something called a Cardiger syndrome, which is an autosomal recessive dynein arm defect, which leads to immotile cilia. Patients come to the emergency department or the hospital with situs inversus, chronic sinusitis, and bronchiectasis. Patients usually end up at the hospital because of frequent respiratory infections, frequent sinus infections, and ear infections, as well as chronic nasal congestion and infertility. The X-ray in this image shows us situs inversus. As you can see here, the heart is entirely shifted to the right side. Now moving on to the sodium-potassium pump. Now the sodium-potassium pump system moves sodium and potassium ions against large concentration gradients. It moves 2 potassium ions into the cell where potassium levels are high and pumps 3 sodium ions out of the cell and into the extracellular fluid. In doing so, it pumps the 3 sodium ions out of the cell. Now, sodium-potassium ATPase is located in the plasma membrane with ATP site on the cytosolic side. For each ATP consumed, 2 potassium go into the cell and 3 sodium go out of the cell. A nice way to remember this is 2 strikes, K, you're still in. 3 strikes, nah, you're out. Now, cardiac glycosides, digoxin and digitoxin, directly inhibit this sodium-potassium ATPase, which ultimately leads to increased cardiac contractility. Alright, moving on. Now we have collagen, which is the most abundant protein in the human body and is extensively modified by post-transitional modification. And this organizes and strengthens the extracellular matrix. Now mainly we have 4 types: type 1, type 2, type 3, type 4. Type 1 is the most common, and it's made by osteoblasts in the bone and can be found in the skin, tendon, dentin, fascia, cornea, and it also present in late wound repair. Type 2 is basically cartilage, including hyaline, and this is found, you guessed it, in the cartilage. Type 3 is found in the skin, blood vessels, uterus, and fetal tissue, and is also a part of early wound care. If there's a mutation in type 3, this leads to a Ehlers-Danlos syndrome and the vascular type of this disease. Type 4 is found in the basement membrane, or basal lamina. So type 4 under the floor, so basement membrane. And this is defective in Alport syndrome and targeted by autoantibodies in Goodpasture syndrome. To remember each collagen and where they're found, remember scab. The first letter of the word being type 1, 2, 3, 4. So type 1, skeleton, type 2, cartilage, type 3, arteries, type 4, basement membrane. Now let's move on to the synthesis and structure of collagen itself. So the first thing that happens is synthesis, which is the translation of collagen alpha chains, or pre-pro collagen, consisting of repeated sequences of gly X and Y, X being proline and Y being lysine. Now keep in mind that hydroxyproline is used for lab quantification of collagen. The second step is hydroxylation. Now the most important thing about hydroxylation that it requires vitamin C. So deficiency in vitamin C leads to scurvy, which is characterized by bleeding and swelling of the gums. Next step is glycosylation. Now glycosylation of pro-alpha chain hydroxylate residues and formation of pro-collagen via hydrogen disulfide bonds. Now this is where the triple helix of the collagen bond is formed. Now if there are problems in the formation of the triple helix of collagen, you can get a disease called osteogenesis imperfecta, which is a genetic disorder leading to the imperfection of bone formation. This leads to people with this condition having brittle and weak bones. Next is exocytosis, where the pro-collagen is ejected into the extracellular space, from where proteolactic processing takes place, which is the cleavage of disulfide-rich terminal regions of pro-collagen, leading to insoluble pro-collagen. Finally, we have cross-linking, which is the reinforcement of many staggered and this reinforcement of staggered tropocollagen molecules are helped with the enzyme copper-containing lysosyl oxidase to make these collagen fibrils. Cross-linking of collagen increases with age. Now, if there's a problem in this part of collagen synthesis, or problems with cross-linking, Menkes disease is usually diagnosed, which is an X-linked recessive pattern leading to failure to thrive, seizures, hypotonia, lack of muscle development, and poor heart control. There is no cure, however. Daily copper injections have found to improve the outcome of this disease. Alright, here we have the different disorders or syndromes involved in problems of the synthesis or the formation or function of collagen. The first being osteogenesis imperfecta, which is a genetic bone disorder, so also called as brittle bone disease, and which is caused by a variety of gene defects, most commonly by COL1A1 or COL1A2. And the most common form is the autosomal dominant, which leads to the decreased production of otherwise normal type 1 collagen. So there's an altered triple helix formation. The manifestations include multiple fractures and bone deformities after minimal trauma, for example during birth, blue sclera due to the translucent connective tissue over the choroidal veins, I can see here. Some form have tooth abnormalities, including opescent teeth that wear easily. So you can say this as dentogenesis imperfecta, and also has conductive hearing loss, which is due to the abnormal formation of ossicles. Now, osteogenesis imperfecta may be confused with child abuse. Now, the treatment is with biphosphonates to reduce risk of fracture. A way to remember osteogenesis imperfecta is through patients can't bite. Bite for bones or multiple fractures, eye for eye or blue sclera, teeth for dental imperfections, and ear for hearing loss. Now we have Ehlers-Danlos syndrome. This is caused by faulty collagen synthesis causing hyperextensible skin or hypermobile joints and tendency to bleed easily or easy bruising. There are multiple types, however, and inheritance and severity vary. It can both be autosomal dominant or recessive, and may be associated with joint dislocation, barium and aortic aneurysms, and even organ rupture. Now, the hypermobility type or the joint instability, is the most common. The classical type, which is joint and skin symptoms, are caused by mutations in the type 5 collagen. Vascular type, or fragile tissues including vessels, aorta, muscles, and organs, that are prone to rupture, and this is mutations in the type 3 procollagen. Now, Menkes disease is an X-linked recessive connective tissue disease caused by impaired copper absorption and transport due to defective Menkes proteins, ATP7A, A for absent copper, or versus ATP7B, which is found in Wilson's disease, and this is copper buildup, which is a whole different disease itself. And this Menkes disease leads to a decreased activity of lysosyl oxidase. And for this enzyme to work, copper is a necessary cofactor. This whole enzyme leads to defective collagen, which results in brittle, kinky hair, growth and developmental delay, hypotonia, and increased risk of cerebral aneurysms. Treatment, like we discussed earlier, is the possible injections of copper. Alright. Now here we have elastin. Now elastin is a stretchy protein within skin, lungs, large arteries, elastic ligaments, and vocal cords, and other organs and tissues. Now this is rich in non-hydroxylated proline, glycine, and lysine residues. Now cross-linking takes place extracellularly and gives elastin its elastic properties. Now this is broken down by elastase, which is normally inhibited by alpha-1 antitrypsin. Now, alpha-1 antitrypsin deficiency results in unopposed elastase activity, which can cause COPD. COPD standing for chronic obstructive pulmonary disease. Now, elastin changes with aging. There's a decreased dermal collagen on elastin and decreased synthesis of collagen fibers as we age, whereas cross-linking remains normal. Now, Marfan syndrome is an autosomal dominant connective tissue disorder affecting skeleton, the heart, and eyes. FBNI or FBN1 gene mutations on chromosome 15 results in defective fibrillin 1, a glycoprotein that forms a sheath around elastin sequencers TGF-beta. Now the findings in these patients are usually tall with long extremities, chest wall deformities, either pectus carnatum or pigeon chest, or pectus excavatum. Hypermobile joints, long tapering fingers and toes, cystic medial necrosis of aorta, aortic root aneurysm rupture or dissection, the most common cause of death, mitral valve prolapse, and increased risk of spontaneous pneumothorax. Now, homocystinuria has similar presentations to Marfan syndrome with pectus deformity, tall stature, increased arm-height ratio, and decreased upper-low body segment ratio, tapering fingers and toes, joint hyperlaxity, and skin hyperelasticity and scoliosis. The main differences between Marfan syndrome and homocystinuria is that the inheritance is autosomal dominant in Marfan syndrome, and homocystinuria is autosomal recessive. In homocystinuria, you have a decreased intelligence. The vascular complications is thrombosis, and lens dislocation is downward, whereas in Marfan syndrome, intellect is normal, vascular complications are found in aortic root dilation, and lens dislocation are upwards, or Marfan's fans out. Now moving on to the biochemistry laboratory techniques. The first one being PCR, or polymerase chain reaction. And this molecular biology lab procedure used to amplify a desired fragment of DNA, and is useful as a diagnostic tool for many viruses and diseases. The most famous these days is for the COVID vaccine, the COVID virus. 3 phases of the PCR takes place. The first being denaturation, which is DNA being heated at 95 Celsius to separate the strands. From there you have annealing, which is the sample is cooled to 55 Celsius. DNA primers, a heat-stable DNA polymerase, TAC, and deoxynucleotide tryptophans, DNTPs, are added. DNA primers anneal to the specific sequence to be amplified on each strand. Then you have elongation, which is the temperature is increased to 72 Celsius. DNA polymerase attaches to DNTPs to the strand and replicate the sequence after each primer. Heating and cooling cycles continue until the amount of DNA is sufficient. So the first step is denaturation at 95 Celsius, then annealing, which happens at 55 Celsius, adding of DNA primers and dNTPs, and then finally is elongation, where the temperature is 72 Celsius and DNA polymerase attaches these dNTPs to the strand, and this repeats over and over again until the amount of DNA is sufficient. Moving on. Now we have Crispr, or Cas9, and this is a genome editing tool derived from bacteria, and consists of a guide, or gRNA, which is complementary to our target DNA sequence, and an endonuclease, or Cas9, which makes a single or double-stranded break at the target site. So you can see here. Now the break is perfectly repaired by non-homologous end joining. Now if any accidental frame shift mutations or knockout mutations or donor DNA sequence can be added to fill in the gap using homology-directed repair, or HDR. Now this is not used clinically, but potential applications include removing virulence factors from pathogens, replacing disease-causing alleles of genes with healthy variants, and specifically targeting tumor cells. Next, we have blotting procedures. And blotting is the process of separating biological macromolecules by size, usually through gel electrophoresis, and then transferring them to a solid membrane, where scientists can detect molecules of interest. So now we have 4 different types of blotting. First is southern blot, where, for example, a DNA sample is cleaved into smaller pieces, which is separated on a gel by electrophoresis and then transferred into a filter. This filter is exposed to radio-labeled DNA probe that recognizes and anneals to its complementary strand. Now, this resulting double-stranded labeled piece of DNA is visualized when filter is exposed to the film. Northern blot is similar to southern blot, except that an RNA sample is electrophoresed, used in studying for mRNA levels, which are reflective of gene expression. Third one is western blot. Sample proteins is separated via gel electrophoresis and transferred into a membrane. Labeled antibodies used to bind to relevant proteins. Now, southwestern blot identifies DNA binding proteins using labeled double-stranded DNA. To remember each procedure of blotting and what they look for, remember snowdrop. SN for southern and DNA, northern for RNA, and W western for protein, so snowdrop. Alright, moving on. Now here we have flow symmetry, and this is a laboratory technique to assess size, granularity, and protein expression of individual cells in the sample. Here, the cells are tagged with antibodies specific to surface or intracellular proteins. Antibodies are then tagged with a unique fluorescent dye. Sample is analyzed one cell at a time by focusing a laser on the cell and measuring light scattering and intensity of fluorescence. Now, this is commonly used in the workup of hematologic abnormalities such as leukemia, paroxysmal nocturnal hemoglobinuria, fetal RBCs in pregnant person's blood, and immunodeficiency such as HIV. The data is either plotted into a histogram or a scatter plot, like right here. Then we have microarrays, and this array consists of thousands of DNA oligonucleotides arranged in a grid or on a glass or silicone chip. The DNA or RNA samples compared are attached to different fluorophores and hybridized to the array. The ratio of fluorescent signal at a particular oligonucleotide reflects the relative amount of hybridizing nucleic acid in the two samples. This is used to compare the relative expression of genes in two samples, and this can detect single nucleotide polymorphisms, SNPs, and copy number variants, or CNVs, for genotyping, clinical genetic testing, forensic analysis, and cancer mutation and genetic linkage analysis. Then you have enzyme-linked immunosorbent assay, or ELISA. This immunologic test is used to detect the presence of either a specific antigen or antibody in a patient's blood sample. Detection involves the use of an antibody linked to an enzyme, adding substrate, reacts with enzyme, producing a detectable signal. And this can have a high sensitivity and specificity, but is less specific than Western blot, often used for the screen for HIV infection. Now here we have on the last page is karyotyping, which is a process by which photographs of chromosomes are taken in order to determine the chromosome complement of an individual, including the number of chromosomes and any abnormalities. Now, colchicine is added to cultured cells to halt chromosomes in metaphase. Chromosomes are stained, ordered, and numbered according to morphology, size, arm length ratio, and banding pattern, which you can see on this picture A. And this can be performed on a sample of blood, bone marrow, amniotic fluid, or placental tissue. And this is used to diagnose the chromosomal imbalances, for example, trisomies and sex chromosome disorders. Now, fluorescence in situ hybridization is where fluorescent DNA or RNA probe binds to specific gene site of interest on chromosomes, as you can see on picture A, and used for the specific localization of genes with direct visualization of chromosomal anomalies at the molecular level. The three possibilities are microdeletion, no fluorescence on a chromosome compared to the fluorescence at the same locus on the second copy of that chromosome, translocation, which is fluorescence signal that corresponds to one chromosome and is found in a different chromosome. So you can see here in the white 2 arrows in the picture A, which shows the fragments of chromosome 17 that have translocated to chromosome 19. And then finally, duplication, a second copy of a chromosome resulting in a trisomy of tetrasomy. So the 2 blue arrows here, showing us the duplicated chromosome 8 resulting in tetrasomy. Then finally, we have molecular cloning, which is the production of recombinant DNA molecule in a bacterial host. And there are 5 steps to this. Number 1 being isolating the eukaryotic mRNA, adding reverse transcriptase to produce complementary DNA, and then insert this cDNA, as it lacks introns, into the bacterial plasmid containing antibiotic-resistant genes, and then transforming, or which involves inserting recombinant plasmid into bacteria. And then the 5th step is surviving bacteria on the antibiotic medium producing cloned DNA copies, or cDNA. Alright, that's it for this video. Thank you for listening, and have a great day.